Hello student, our today's lecture is again on Treponema pallidum part 2. This is important spirochetal infection of human being which is included in chapter study of bacterial pathogens. Subject medical microbiology, paper MB 331. In this we'll see in detail remaining part of Treponema pallidum with respect to immunity, laboratory diagnosis, then prophylactic measures, that is prevention of the disease, as well as the treatment and its epidemiology. In case of a treponema, upon infection, immunity develops in the body. Syphilis has persistent infection for decades, in spite vigorous host response due to its dense coat. This dense coat is with fibronectin, transferrin, and ceruloplasmin. There is an inhibition of a cell-mediated immunity. Relative but unreliable protection is observed from reinfection in untreated patients. There is a minor protection from reinfection in treated patients. Basically, upon entry of the treponemas in the body, antibodies are developed, and after primary syphilis, there are less chances of a development of a secondary syphilis if the immune status of the person is better. After development of phase symptoms, that is chancre, condylomata, or in case of a tertiary syphilis, gammas, we have to collect material or the exudative material from the lesions which appear on the skin. Because of inability to culture this Treponema pallidum using standard laboratory methods, direct detection methods play important role. That is, after collection of a samples, that is exudative material from the lesions, open lesions present on the body, one can stain them by using various techniques. This is most reliable method. Direct detection method, that is by isolating microorganism in the laboratory, is not preferred method. It requires specialized laboratories as well as the treponemas from clinical samples unable to grow in the media like Novochi's media. So dark field microscopy is widely used for laboratory diagnosis. It is most sensitive method as well as it is direct and a quick method for diagnosing syphilis in the active lesions. Third test which can be carried out in the laboratory diagnosis is RT-PCR. Here the genetic material of treponema, they are identified in this test. So here you can see one chart showing two major types of a test, non-treponemal test and the confirmatory treponemal test. Non-treponemal tests are non-specific tests because as this organism is not cultivable one, different types of antigens which are not from treponema origin, they are used for the testing. So they are called as a non-specific test. So one important test included in this non-treponemal test category is VDRL. VDRL, Venereal Disease Research Laboratory Test. And the second one is a Rapid Plasma Reagin Test or RPR. Here, this non-treponemal test detect antibodies that are not specific to Treponema pallidum, so-called non-treponemal test. While in the second category, which are confirmatory treponemal test, Treponemal pallidum particle agglutination test, that is TPPA, as well as fluorescent treponemal antibody-absorb test that is FTA-abs, Treponema pallidum enzyme immunoassay antibody test, that is TPEIA, and lastly chemiluminescence immunoassay, that is CIA, widely used as a confirmatory treponemal test. These tests are based upon the detection of antibodies directed against specific treponemal antigens, and so they are important in the diagnosis. Treponemal tests are qualitative only, that is, one cannot measure quantity of organisms or antibodies present in the patient's blood. So qualitative, just they detect whether the antibodies against treponema are present or not. They are respond as a reactive or a non-reactive and not quantitative one. Immunofluorescent and immunoperoxidase antibody staining is also performed in the laboratory diagnosis in the detection of treponemas. These specific immunofluorescent or immunoperoxidase antibody staining can be visualized to use that organisms, including non-viable spirochetes from mucocutaneous lesions as well as a lymph. As you know, during infection, mucocutaneous junctions, on that there is a formation of a lesions. So gammas, condylomatas, they are rich in microorganisms and one can collect material from it as well as in the patient's blood, that is in the sera, antibodies are present. So that can be stained here. Serological testing play important role in the diagnosis. The first test is called as a Treponemal test, which uses syphilitic tissue as a complement fixing antigen, while second one uses one specific strain of Treponema called as a Reiter strain. This is non-virulent treponeme, and from that antigen is extracted. So name given to the test, Reiter Protein Complement Fixation test, and here it is non-reactive in late stages of syphilis, so not widely used for the laboratory diagnosis. In the serological testing, as you know, non-treponemal tests are included like VDRL and other complement fixation tests. The antigen used here is cardiolipin lecithin antigen. So here non-treponemal test where antigen treponema is not used, but instead of that some other antigen which is not specific is used as an antigen. So complement fixation test like Wassermann and Kolmer is used by using this cardiolipin lecithin as antigen. And second and important test which may give false positive result is flocculation test. So VDRL, Venereal Disease Research Laboratory test, and Hinton Rapid Reagin test are included in this non-treponemal test. Non-treponemal reagin antibody test. So here syphilis reagin antibodies are IgG and IgM antibodies. They are directed against lipoidal antigen resulting from the interaction of host tissues with Treponema pallidum. This is the earliest cardiolipin antigen used to measure reaginic antibodies. They were crude extract from beef livers or a beef heart, and they may show false positive test. Again, I'll repeat this point, that is, this test is not specific because instead of a antigen from Treponema pallidum, use of a beef heart or beef liver, which is cardiolipin antigen used for testing. Means in the patient's blood, antibodies are present and they react with this cardiolipin antigen from beef liver or a beef heart. So non-specific type of a non-treponemal reaginic antibody test. Nowadays, Use of a cardiolipin cholesterol lecithin. This one is much purer and hence there are a less false positive reaction. The non-treponema test first developed was VDRL slide test, and in this serum is heated to 56 degrees Celsius and then tested to see if it can flocculate a suspension of a cardiolipin cholesterol lecithin antigen. Again, the original treponemas or the exact treponemal suspension is not used as a antigen for VDRL. Except for its use in diagnosing neurosyphilis, these tests now been largely replaced by other tests like rapid plasma reagin RPR card test or ART, that is automated reagin test. The important test which can be used for confirmation purpose is fluorescent treponemal antibody absorption test, FTA-abs. This FTA-abs uses Treponema pallidum harvested from rabbit testes as the antigen. So they are specific. Direct visualization by dark field microscopy, where the fluorescent antibody which are nothing but present in the body and they are treated with the fluorochrome. So special stains are used for this purpose, which is called as a Lyophilized Nichols strain organism as an antigen. They binds to the antibodies present in the patient's blood and they fluoresce in this way. So here fluorescent treponemal antibody absorption test is important in the confirmation of a syphilis. Next test is enzyme-linked immunosorbent assay, that is ELISA. The syphilis ELISAs are automated tests that use a technique called as a qualitative sandwich immunoassay. This is to detect treponema, again Treponema pallidum specific antibodies. Not only this, with this one can use animal inoculation test for the laboratory diagnosis. And here rabbit models are always a preferred one. For the development of trials for vaccine development, for the vaccine trials also, rabbit models are used widely. Last part is of treatment, prevention and of epidemiology. Now, here in the treatment part, penicillin is the drug which is widely used to treat first and second stage, that is primary syphilis and secondary syphilis. There is a complete recovery is observed in the patient upon treatment of this. So this is again unique characteristic of a treponemas, that they are considered as a gram-negative microorganism, but then also penicillins, which are normally used to treat gram-positive infections, they are used here and they show better results. With that, some broad-spectrum antibiotics like streptomycin, tetracycline, erythromycin, they are used in the treatment of the syphilis. But as there is a poor passage of these drugs to fetal circulation, there are less chances of use of such a broad spectrum antibiotics in case of a pregnant woman. Now about prevention of the disease, that is prophylactic measures. So as per CDC, in 2018, last trial on the development of a vaccine against this, and it is observed that whatever vaccine prepared by using a rabbit model not at all effective for prevention of the disease. Several vaccines based on treponemal proteins, they reduce lesion development, that is gammas, the tertiary stage symptom lesion developed during infection. This is again in animal models, and nowadays research continue on the development of a better vaccine for human beings. The most effective prophylactic measure against contracting this syphilis is avoidance of a sexual contact with person who harbor the spirochete. Treatment to case contact also one way to control a disease. So to the cases of syphilis less than 2 years of a duration, one dose of a benzathine penicillin 1.8 gram by intramuscular injection is required, and if they treated 100% recovery is observed, and it helps in the prevention in the spread of disease from one person to other person. So here we can call it as a not exactly chemo prophylactic measure to the other members of the society, but proper treatment to the cases. In the epidemiology part, we have to see spread of the disease in various groups, in the various races, as well as here the countries which are mainly affected. So syphilis can be spread by passage through the birth placenta, that is congenital syphilis, or other close contact with an active lesion. Also, blood transfusion may lead to syphilis in the individual which are recipients one. Then direct inoculation, this is mainly a STD type of a disease. So the vast majority of cases are transmitted by sexual contact. This disease is worldwide in distribution, and in 2016 more than 88,000 cases of syphilis were observed according to the CDC, that is Center for Disease Control and Prevention. The rate of women with syphilis has been declining in the United States, but the rate among men, particularly men who have sex with men, has been rising, that is homosexuals. It occurs primarily in persons between the ages of 15 and 40 years, but about 1 million infants are born each year with congenital syphilis. So this one is a quite serious problem, that is from pregnant women suffering from syphilis transmission to their fetus. While syphilis infection is easily identifiable one and treatable one, rates of syphilis infection continue to increase among the populations in high-income countries, and it remains at endemic levels. So in the epidemiology, we have to mention that worldwide this is endemic type of a disease, that is few cases continuously observed in the population. This is mainly in low and middle income countries, endemic type of a disease, syphilis. So we can summarize this part of Treponema, that CDC approved standard test including VDRL slide test, then RPR test, as well as an untreated serum reagent test. Nowadays, one more test is included called as a toluidine red unheated serum test. It is also called as a TRUST. Then some non-treponemal tests, they are also widely used. They are rapid, simple, inexpensive, but cannot be used for the confirmation of the disease. For that, direct methods require, like dark field examination, and these tests are used to detect Treponema pallidum directly from whatever symptom, whatever the lesion present on the patient's body. So here the lesion exuded, material from that it is collected, or the tissues affected, they are by biopsy method they are collected, and then they are diagnosed by using direct microscopic observation. So this is about laboratory diagnosis. I can summarize in the treatment part, that is penicillin G is a promising drug. It is administered parenterally, that is in the injection form, and is preferred drug for treating persons of all stages of a syphilis. So this is all about Treponema pallidum. Thank you very much.